Good afternoon, everyone. I'm delighted to welcome you. This is September 11th, which is a somber day of reflection on the tragic loss of life in New York, Pennsylvania, and Washington, D.C. back in 2001. It's also a day for life affirming looking to the future. And in that spirit, I was delighted to invite my good friend, Dr. Eli Adashi, to speak to us on in vitro gametogenesis. Eli's bio attests to why he is the best possible teacher for this fascinating, if not mind-boggling topic. Eli obtained his MD degree in Tel Aviv University, his master's at Harvard in Public Health and Healthcare Management, and went on to training in obstetrics and gynecology at Tufts. He then did an reproductive endocrine fellowship at Johns Hopkins and reproductive biology training at UCSD, and then joined the faculty at the University of Maryland, and then on to the University of Utah, and then to Brown in 2004, where he served as dean from 2004 to 2008. Eli was funded by the NIH for approximately 20 years and became really one of the leaders in reproductive biology. He's a member of the National Academy of Medicine and has served on many important committees, including the one on human embryonic stem cell research and women's health research. He is an extraordinary expert in reproduction, in science, in law, ethics, and human rights. And his works have appeared not only in the scientific literature, in numerous papers and books, but also in the New York Times, the Washington Post, and the Boston Globe. He also has played a role at NIH at the national level, serving on the Council of the National Institute of Child Health and Human Development. Among his many important ongoing activities is chair of the Medical Executive Committee and the Medical Advisory Council of the Jones Foundation for Reproductive Medicine, the Advisory Council of the Hastings Center in Bioethics, the Board of Directors of Physicians for Human Rights, and he's been a key advisor to the World Health Organization and the Bill and Melinda Gates Foundation. He really is one of the nation's leaders as an advocate for women's rights, as well as the rights of women around the world. He last spoke here in the same room when we had a symposium on mitochondria. And at that time, he spoke about mitochondrial repair. And I think that was a fascinating discussion. And we are looking forward to an even more fascinating and both medically, scientifically, and I would say ethically challenging topic of in vitro gametogenesis.
Thank you very much, Barry. Thank you all for sacrificing your lunch and or 12 o'clock break to come and listen to this presentation. I wrote to Barry in advance when I realized that it was 9/11, and I asked his permission to say a few words, in that I couldn't see just moving on without at least making a mention of the tragedy. I don't have to tell this audience that on 9/11/2001, at 8:46 a.m., a commercial airliner, which was hijacked by Al-Qaeda terrorists, crashed initially into the North Tower of the Twin Towers of the World Trade Center complex. And not too long thereafter, at 9:03 a.m., the South Tower of the World Trade Center was similarly struck. Both 110-story skyscrapers collapsed, of course, shortly thereafter, thereby bringing about the destruction of the remaining 5 structures in the World Trade Center complex and damaging and destroying nearby buildings. But most importantly, close to 3,000 people lost their lives and thousands more were injured. I will simply say, and I know you will agree, that we will never forgive nor forget. From that to in vitro gametogenesis is a very sharp turn and a somewhat awkward one. But we are, after all, here to advance our collective insight, and that's what we're going to try to do today against the backdrop of these very sad memories. This audience doubtlessly knows that the process of in vitro fertilization, which is the last hope for women with the type of infertility that does not lend itself to other approaches, was a brainchild of the late Robert G. Edwards, PhD, who accomplished the first IVF pregnancy ever in 1978 in the United Kingdom. That accomplishment was rewarded by the Nobel Prize in Biology and Medicine in 2010, solely to Dr. Edwards, simply because his 2 collaborators have passed on by then. And the reality was that Dr. Edwards himself, at that point, was too compromised himself to actually receive the prize. It was his wife who ended up flying to Stockholm and bringing home the award. But the requirement, as you know, of the Nobel Prize Committee is that the winners be alive at the time of the award, and he was technically alive, but really unable to either appreciate the award, let alone receive it. But that was a landmark that changed reproductive history in ways we could not have envisioned before. Staying away from overstatement, it is nevertheless tempting to consider the possibility that in vitro gametogenesis is perhaps the next step in this evolution. The notion that you could ultimately create an egg or a sperm from one of your skin cells is in itself rather difficult to fathom and or digest. And yet this is exactly what in vitro gametogenesis is all about. Although we will not dedicate a great deal of time to this today, there is a separate conversation I usually carry on that. What in vitro gametogenesis also makes possible, which IVF could not, is establish same-sex parenthood. That is to say, 2 individuals who cohabitate, who are of the same sex, who invariably are unable to establish a biologic pregnancy, now are in a position to do so, or will be in time in a position to do so, because the tools and the knowledge and the preliminary data are now here to suggest that 2 men could potentially be the genetic parents of a child, although they will need a surrogate, of course, to carry the pregnancy. And 2 women could be the genetic parents of a child. These are couples who are in a same-sex relationship. I speak about that separately because this is an important topic and one that takes us a step beyond where we will be today. But I mention it simply because it's breathtaking in so many ways. Just the very notion that same-sex couples could be the genetic parents of their children. It is probably somewhat difficult to pinpoint the beginning of this science. I chose, but with no special conviction, a Cell paper from 2009, senior authored by Professor Mitinori Saito from Kyoto University, titled A Signaling Principle for the Specification of the Germ Cell Lineage in Mice. And I picked one statement from that paper, reading, germ cell fate in the epiblast is a direct consequence of BMP4 signaling from the extraembryonic ectoderm. This was really just a very, very early step in defining the pathway that takes a primordial cell towards a mature germ cell. And Mitinori Saito was and remained a key player in this arena. He is joined, or has been joined, by Katsuhiko Hayashi, PhD, who at the time I visited with a couple in 2013 or so in Japan, was probably a postdoc or a fellow or an associate of Dr. Saito. He has since moved on and established his own laboratory in Kyushu University. But anywhere you look in vitro gametogenesis to this day, has in one way or another the imprimatur of Professor Saito and Professor Hayashi. Both are exceptionally capable investigators and have really broken a field that did not exist simply before. The experimental paradigms we will try to touch upon today will involve the rodent and the human, and will emphasize the fact that all of this has everything to do with pluripotent stem cells. And when we say pluripotent stem cells, we could mean either embryonic stem cells or induced stem cells, but either way, this is the starting point from which Hayashi and his colleagues have taken the process to the mature gamete stage. Now, pluripotent stem cell lineages could be used in a whole host of tissues, of course. If we knew how to do it, we could produce amacrine neurons and place it in the retina in those who genetically lack those. We could conceivably create dopaminergic neurons, and if we knew how, perhaps instill them into the striatum in individuals who are coping with Parkinson's disease. Now, that may seem like a simplistic solution, and it may be, but it's at least a step closer to addressing the dopamine deficiency of that condition. Or, in our wildest dreams, would we potentially produce motor neurons and replace missing motor neurons in the spinal cord and have a paralyzed individual walk again? Now, if all of this sounds like science fiction today, it is. But I think what we are seeing today and where we find ourselves in today is at the beginning of a journey that will allow us to essentially create any cell type in the body if we so chose, in principle, although my focus today would be on the gametes. Specifically, stem cell-derived gametes, and I will, of course, expand on that. The importance of this notion did not escape Science magazine, And in 2021, they dedicated a significant spread to a paper by Professors Saito and Hayashi. The New England Journal of Medicine, not to be outdone, also dedicated an article titled Oocytes from Stem Cells, authored by Mary Herbert and Azim Zourani from the UK. Last year, yours truly organized and chaired a 2.5-day workshop in Washington, D.C., under the auspices of the National Academy of Medicine. And the expectation is that a consensus committee report will follow at some point. If you look at the Nuffield Council for Bioethics, which, as you know, is the premier ethics institute of the United Kingdom, and you look at their annual agenda, which tends to use this particular depiction, you will see that topping the list of the Nuffield Council on Bioethics is in vitro derived gametes in the field of human reproduction. Yes, they will deal with time limits on maintaining embryos in culture, perhaps womb replacements, perhaps preconception screening, whole genome sequencing of embryos and fetuses, genome editing of gametes and embryos, and egg freezing, to name a few. But topping the list, you will notice, or you have noticed, was in vitro gametogenesis, which in so many words is a disruptive technology. The question is it achievable in the human? And we're not there yet. I want to make that very clear. And what is the timeline? In other words, when can we expect it to be achievable in the human? All of this, of course, tempts one to go back to the tried and true, such as Aldous Huxley and his Brave New World, which goes back to 1932.
Great. So back to Aldous Huxley, he made several predictions out of many that I will not list today. He predicted that eggs will be produced via ex vivo ovarian cultures, that embryos will be generated via in vitro fertilization, and that newborns will be created via ex vivo ectogenesis, which deserves a presentation in its own right, although this field certainly still lags behind. Of all those three, he was, of course, dead right when it comes to in vitro fertilization. This is a reality today, and he was able to articulate that notion quite some time ago. Of course, his idea was, and that's in chapter 1 of Brave New World, and a statement by the so-called director of hatcheries and conditioning, who said, one egg, one embryo, one adult, normality, making 96 human beings grow where only one grew before, progress. And it is true that today, when we speak about in vitro gametogenesis, the vision of 96 well plates is everywhere. And so, again, going back to Brave New World brings about some sense of the visionary that the author was. Now, there are some obvious obstacles, of course, to in vitro gametogenesis. And those are mostly theoretical, I should say, because for the longest of time, our views were shaped by the so-called soma germline barrier hypothesis, which was the brainchild of August Weismann, professor of zoology at the University of Freiburg, who articulated those in his 1893 book, The Germ Plasm, A Theory of Heredity. And that book dominated the reproductive thinking for the longest of time. And in it was, of course, the dominant soma germline barrier hypothesis, according to which the germline, which gives rise to soma, which is immortal, which would be our view, he refused to acknowledge the fact that the soma never gives rise to the germline because it is mortal. And of course, in vitro gametogenesis is all about the soma giving rise to a germline. But for the longest of time, conceptually, this was deemed infeasible, impossible, unreasonable by dint of the influence of deep thinkers, such as our colleague. Which brings us to the notion of pluripotent stem cell formation technologies, of which somatic cell nuclear transfer was probably the first. This goes back to 1958 and to the work of John Garden, who in a paper in Nature titled Sexually Mature Individuals of Xenopus laevis from the Transplantation of Single Somatic Nuclei, proceeded to take somatic karyoplasts, inject them into an oocytic cytoplasm, and created what amounts to a zygote, essentially a blastocyst with an inner cell mass. And so from there, he could grow this construct into embryonic stem cells. He was joined in 2006 by the induction technology promoted by Shinya Yamanaka from Kyoto University, whose cell paper in 2006 was titled Induction of Pluripotent Stem Cells from Mouse Embryonic and Adult Fibroblast Cultures by Defined Factors. This was a variation on the theme. He started with a somatic cell and used selected transcription factors that were identified by then to convert the somatic cell into an induced pluripotent stem cell that could then go anywhere. This discovery of a pluripotent stem cell that can be the origins of any cell type we choose if we know how to get there was rewarded by the 2012 Nobel Prize in Physiology and Medicine to these two individuals. But you have to say that this discovery was a leading element of what eventually led to the notion of in vitro gametogenesis. Whether or not this has anything to do with the fact that Professor Saito, who is really the godfather of IVG, is from Kyoto University, I don't know. I can share with you that Kyoto University has an impressive stem cell institute wherein both Professor Yamanaka worked and works, and where, of course, Professor Saito works. But in vitro gametogenesis, to go back to our subject matter, is all about converting a somatic cell, no matter which type, into either an embryonic or induced pluripotent stem cell and then guiding that stem cell towards a gametogenic fate. Now, there are some prerequisites. You can't just do that because the inspiration struck you. You have to know the germ cell specification pathway. In other words, you have to know the cues that the stem cell will require, the sequence by which you will apply those cues, the timing of the application of those cues, and you always have to consider the niche in which these stem cells are operating. After all, as you know, the oocyte is encased by granulosa cell, and the sperm are encased by Sertoli cell. And that's not a coincidence. You cannot develop gametes in thin air. There has to be a somatic cell component, and that's what we define here as the niche. It's a little difficult, as I said, to nail down the development of in vitro gametogenesis or the creation of germ cell elements from stem cells. But a science paper from 2003 titled Derivation of Oocytes from Mouse Embryonic Stem Cell appears to be a strong candidate for this possibility. The first author was Hans Scholler, who is presently the director of the Max Planck Institute for Molecular Biomedicine, but at the time was a postdoc at Penn. And it was this science paper that perhaps inaugurates this new arena. That same year, Toshiaki Noche from Japan reported on the pages of PNAS that embryonic stem cells can form germ cells in vitro. Same year or a year later, George Daly, who is now the dean of Harvard Medical School, reported on the pages of Nature the derivation of embryonic germ cells and male gametes from embryonic stem cells. And René Rejo-Pera, who at the time was at UCSF, reported on the pages of Human Molecular Genetics the same year that about spontaneous differentiation of germ cells from human embryonic stem cells in vitro. The next 5 to 10 years have seen a plethora of papers in leading journals, as you can tell, in this case, describing the germ cell specification pathway in the rodent. A similar sequence that described germ cell specification pathway in the human was likewise apparent and so pressed in the literature from 2009 through 2015. And so the state of the art in 2015, as it stood, was that the germ cell specification pathway was now shown to be not conserved between the rodent and the human. In other words, different actors were involved, and that the genetic networks involved are almost by extension quite distinct as well. Thinking of this more schematically, the state of the art in 2015 was that we could envision embryonic stem cells or induced pluripotent stem cells being converted to primordial germ cell-like cells, but not mature germ cells, in the extragonadal context. But very little was known about the intragonadal process, which took haploid postmeiotic, which produced haploid postmeiotic mature gametes. So the extragonadal pathway was beginning to be delineated, but the intragonadal pathway was still ahead of us. And so the final frontier was what you could call in vitro gametogenesis, 'gametogenesis all the way. Step in our old friend Katsuhiko Hayashi, who in 2016, on the pages of Nature, reported the reconstitution in vitro of the entire cycle of the mouse female germline. Still in the mouse, but all the way. What he did was, he started with embryonic stem cells or induced pluripotent stem cells, converted them to primordial germ cell-like cells, combined them with somatic cells, as would happen in vivo, and thereby produced a primary oocyte, shown on the left-hand side of the slide, which upon fertilization gave rise to an embryo, eventually a blastocyst, which upon transfer to recipient mice gave rise to pups. In a way, the whole process from beginning to end, from stem cells to gametes to pups, albeit in the rodent. Since he was lacking mouse or rodent ovarian follicles, he, and I said this half jokingly as a side project, he proceeded to produce granulosa cells of rodent origin, and that paper saw press in Science in 2021. And so on the female side of the rodent, we now had a sense that we could take a stem cell to a germ cell and ultimately produce a pregnancy. Much of the same was later accomplished on the male side in a paper published in Cell Stem Cell in 2021, this time by Mitinori Saito. Again, he went all the way from induced pluripotent stem cells to sperm, which were then used to fertilize eggs and create pups. So in the rodent, it seemed like we were making good progress, but the human paradigm was still beyond our grasp. Step in, yet again, our old friend Mitinori Saito in 2018 in a science paper titled Generation of Human Oogonia from Induced Pluripotent Stem Cells in Vitro. Now, that was a clear advance, but regrettably, as you will realize, did not take us all the way, but it took us a big portion of the way. He started with human primordial germ cell-like cells, combined them with somatic ovarian cells, and was able to take the process in vitro all the way to the oogonial stage, which is an advanced stage of oogenesis, but is not the same as an oocyte and would not really be fertilizable and or useful if this technology were to be applied in the human, particularly in the infertile human. The same more or less was accomplished in the human male side by Kotaro Sasaki, who by then, I believe, was a faculty at the University of Pennsylvania, where he may have started as a postdoc. So in terms of human in vitro gametogenesis, we have yet to get to the stage where we can, in the laboratory, produce M2 oocytes and or spermatozoa. For example, in the case of individuals who lack them altogether from birth. But they have other somatic cells which we could use to produce gametes, even though they may lack them in their gonads. And so there was a significant scientific and translational process that on the one hand could advance science because we now would have an inexhaustible supply of germ cells. It could allow for functional null mutant analysis, which is critical to define the functional genes. It would allow the study of germ somatic cell interactions, which is key to reproductive function, and clinically speaking, could in principle reverse germ cell failure by autologous gamete replacement, by gene-edited gamete replacement, if such were called for, and as I mentioned at the very beginning, by same-sex genetic parenthood, which would have been, that's the wrong word, inconceivable, would be inconceivable though, really until very, very recently. And even today, as I speak about the subject, I find it mind-boggling, even though I have delved into it deeply on a number of occasions. Now, this will not be without an impact on the clinical practice of IVF. Right now, IVF is a golden standard. It's what we rely upon. But IVG, if it becomes a reality in the human, as we expect it will, in time, it will eliminate the need in stimulation, meaning gonadotropin stimulation of egg production, and also the surgical process of egg retrieval, which currently women have to endure. It will do away with gamete donation, which is currently, of course, a big piece of the infertility action. Will it be age dependent? Most likely, but again, those are open questions at this point. And in essence, what it will do, it will take the IVF of today and convert it to effectively a laboratory procedure. No need in the injection of gonadotropins, in the monitoring of hormone responses, in the surgical retrievals of eggs. All of that will be put behind us, and this will be transpiring in a lab. The only residual step that still will be always required is endometrial preparation and ultimately embryo transfer. But everything prior to that probably will be relegated to history and will no longer be required. Whether or not this will improve the notoriously expensive IVF process and thereby access is a big question in my mind, because we have made this statement many times before in the context of many other advances, and more often than not, we were disappointed. It seems that American capitalism finds a way around all of this, and one way or the other, the price either stays the same or in many cases goes up. So the promise of IVG, although clinically real, is a little bit uncertain when it comes to affordability. Now, I don't need to tell you that none of this can happen in thin air. This has to be subject to significant regulation, and it will be when it is ready in the human. This is a matter for FDA jurisdiction. It will be specifically adjudicated by the Center for Biologics Evaluation and Research, or CBER for short, and will be relegated to what they call human cells, tissues, and products, or HCT/Ps. But there is no question but that whoever comes up with the first commercial product of IVG will have to conduct proper clinical trials that will hopefully prove in every way acceptable, and submit those results for review by CBER at the FDA and the committees that they convene to this end before this can become a clinical reality that is being applied to people. Which brings us full circle to the Nuffield Council on Bioethics, which has placed in vitro gametogenesis at the top of his agenda. And I happen to agree with them that this is a worthwhile effort to invest in. It is something that will take some time to definitely clean up, finish up, materialize, approve. But while some of us in this room may not be around when IVG becomes a reality, I have little doubt but that IVG will have a profound effect on human reproduction for those of us who are unable to reproduce naturally on our own, whether it's because we are plagued by infertility or whether or not we are engaged in a same-sex relationship. So IVG, to my way of thinking, is yet another element in the mix that is added on top of IVF, to some degree will replace IVF, but will be the name of the game, particularly of the young members of the audience, who will either see to its realization, potentially experience it if appropriate, and on the whole, be there to see it materialize in real life. This will essentially mean gametes to order, which I don't view negatively in the context of people we are trying to help to conceive. And so for now, all I can say is stay tuned. I find myself often having to say that because we're picking on topics while they evolve, as opposed to discussing final products. There is no final product here, and there may not be for some time to come. But eventually, I am convinced that this will be a material and fundamental change in the field of reproductive medicine and the applications thereof. So with that, Barry, thank you very much for the opportunity, and thank you all for spending this precious time around midday to listen to this aging professor blabber around.
Well, I'd say mind-boggling was the word I chose, and I think the more I hear, the more mind-boggling it is. So congratulations to you and for sharing it with us in both the historical and the scientific method. We're open for discussion. Anyone have any questions? Sarah and then Matt.
And then the second question relates to that question. Is that, as you know better than I and the rest of us, reproductive medicine is really almost exclusively in the, it's not very well regulated, right? It's within the, I don't know, private practice and academic medicine. It's not a government activity for the most part. And there are lots of organizations that guide us, but in some places like China, what's going on?
Yeah, thank you. So I think your first question had to do with the timeline, I believe. And of course, if I studied better, or listened better in my Bible class, I could have quoted one of the standard statements here, namely that the future is, of course, unforeseen, and the pace of progress is unpredictable. And so I don't really know that we can make any even quasi-sound predictions. All I can say is I am continuously, thoroughly impressed by the people who are driving this, specifically the two Japanese investigators I mentioned. This is based mostly on really their published output and their accomplished efforts, but to some degree also on my personal assessment of who they are by dint of having met them and having stayed in touch with them. And when they're in the United States, I make an effort to visit with them and invite them to conferences that I may be involved with. Because I think overlooking that facet of reproductive evolution is like putting blinds over your eyes, because this is the future. And when and if so, that's where I think we're struggling. So no, I really can't give you a timeline. When you consider that the process is not scientifically concluded, the process has to still undergo clinical trials in the US. The process will have to be approved by the FDA. I mean, we can only venture a guess. I don't think we can make any intelligent projections. And your second question was regulation.
Well,
Regulation.
and what's going on, say, in China, where there's
Yeah.
so much less.
Yeah,
Yeah.
I wish I could say something intelligent about China. I visited there several times in an effort to educate myself. But I think it's evident that the size of the country and the nature of the governance doesn't lend itself to the same kind of intensity that we are used to. That's not to say that there is no FDA equivalent in China. That is not to say that the Chinese are in some way irresponsible or proceeding without evidence. I think they will doubtlessly contribute at some point in the process, because the resources are, from our point of view, all but infinite. So many universities, so many investigators, so many resources ultimately, that I have to believe that China will continue to be a world player. And if I had to guess, an ever more important world player. I mean, we can bring 300 million people to the table, but the Chinese can do much better than that.
Thank you.
Matt? I'm curious how you or others in the field are thinking about a potential hurdle or potential problem in particular. What people have observed that throughout life you acquire somatic mutations, for example, in skin, esophagus, any other tissues. It's easy to imagine that if you pick a cell with the wrong mutation, suddenly you've caused, say, a P53 mutation which accumulates. You've caused, I believe, from many in the child. You could imagine bypassing sort of physiologic restrictions on mutations, where some mutation in vivo wouldn't go through the whole process to make a germ cell, but suddenly in vitro you can bypass this and you've created a new human disease. On the other hand, you could argue that maybe by accelerating sort of germline genetic diversity, you could somehow enhance evolution in some way. It's complex, but I'm curious what's the thought about all of it.
Thank you. To my mind, this is well thought out, very apropos, and not to be dismissed. We do have to be mindful of what somatic cells, for example, we bring into play. And some of it may be known from the systemic circumstance of the individual from whom those cells are derived. In general, you probably want to focus on younger members of the species who don't quite yet have the same opportunity to um, to lose the, shall we say, the purity of that comes with birth. But I think not paying attention to the point you raise, I think, is using blindfolds and is wrong-minded. And because the last thing we want here is to cause more harm to individuals who have already paid a dear price in terms of heartache and just the very challenges of life without children for those who wish to have them. So your point's very well taken, and I think some technology would have to be made possible. And as I said, preference should be given to as young a resource as possible, such that acquired mutations are at least kept to a minimum. You cannot assure, likelihood the complete absence of those. But clearly, the younger we are, the better off we are. Yeah, sure.
There was a study recently in Nature Medicine that showed the IVF conceived children have shorter telomere length compared to spontaneous
I'm sorry.
conceptions.
That IVF, children born of IVF have shorter telomeres.
Oh,
And that the longer they were, so like they were cultured, I think, day 3 versus day 5, and the day 5 study even shorter compared to day 3s compared to the spontaneous conceptions. And I think that just gets to the theme that, you get to the birth of the child, but there's the whole life that they have ahead of them that we don't know, like what the consequences of everything that happened upstream are going to be in terms of epigenetic changes, other, non-genetically, like phenotypes.
Is there evidence of IVF children having shorter lifespans or tissue burn out faster? Or I guess the implications of the shorter telomeres.
well, I would say two things. First of all, a very appropriate concern, and one that need to be taken into account in what we just said, namely that this cannot be introduced willy-nilly into the population. This will have to undergo serious, credible evaluations, as I said, by the FDA ultimately, although long before that, there'll be multiple layers of review and assessment as well. I think there was an additional question there.
Do you have a question?
No.
So
But wait, did you have another question besides that concern?
No.
No, okay.
So let me, I want to ask one really scientific question, and then I just want to make two comments. So let me start with the comments first. The first is that IVF isn't regulated in the United States, but it's regulated in the UK. And I think the United States, I'm not talking about the FDA regulation now, I'm talking about the practice of doing IVF. And that's a big problem. And I learned about this being on the National Academy's report or committee on human genome editing. So we really have the Wild West in this country about reproductive technologies, and that's a choice that we can reconsider. And frankly, I think we should have reconsidered a long time ago with legislation, and I think should still be very seriously considered. The other thing I wanted to say, and I deeply appreciate your focus on the parents who have challenges with regard to fertility, but it's impossible for me to sit here and not wonder about what I'll call the parentless child, where the government decides that they want a lot of children and simply take skin cells from young children that they may gain access to in any one of a million ways, and start making humans that have no parents but are wards of the government. We're back to Huxley, but this is not at all inconceivable. And in a world where people are obsessed with immigration and nationalism, as well as declining fertility rates, I just want to say that regulation here and societal discussion and understanding all of the implications, including the possibility that you can now produce a human who would essentially be haplo, right? What would a haplo human look like? So those are all of, you can see where my head is. And I apologize for that, but I think it's good to really broaden this discussion to understand just why mind-boggling, I think, is the right word. But let me come back to the science, because I think the science is really interesting. So what is so challenging about meiosis? Because that seems to be the last step here that is the big stumbling block. So why have the rodent meiosis been able to be achieved, but not the human?
Good question. I'm not actually sure I know the answer to that, because the same very able investigators are struggling with both. You've just stimulated me to write to them and ask them, what in the world is holding you up so much on the human side, whereas you seem to be steamrolling through the rodent side. But I think in a more serious vein, it's not as if the rodent is any less complicated than the human. But first of all, I would say they started doing it much later. So the initial effort, rightly so, I think was focused on the rodent. Now they're beginning to understand that it's time to move to the human, and so they're doing so. But I've shown in the slides that I've shared today the limits that they have been able to reach, and to recognize that there's still a ways to go. So, but again, just my deep admiration and appreciation of the ability of the individuals involved suggests to me that you shouldn't fall off your chair next week if you get a science magazine which spells out the entire sequence in the human. But they don't publish anything that is not fully vetted, fully worked out. And when they do take it to a certain point, they point exactly where the handicaps are, where the issues are, where the limitations are. And so I have great, really confidence in their ability, in their moral character, in their ethics. When they have it right, when they know what they're doing, we will hear about it.
We're right at the hour, so I want to again thank you for expanding our mind, presenting us with the challenges we need to face going forward, and the opportunity.